Welcome back, everybody. Hope you had a great break. So I'm excited to announce our newest kid on the block to join us today. Michelle and I went out to their offices out in San Francisco this past year, and what an honor. We got to speak to their entire organization and really just share what hypoparathyroidism is about, and again, to help them understand their why. And I mean, they couldn't quit raising their hand with the different questions, so it was really very exciting. So we appreciated that opportunity. Today, speaking for Septerna, we're going to have board-certified cardiologist, Chief Medical Officer at Septerna, Dr. Jae Kim.
Thank you. Thank you, Patty, Michelle, Bob, and the Hypoparathyroidism Association for the kind invitation for Septerna to come here today. My name is Jae Kim, Chief Medical Officer at Septerna, and I'd also like to introduce you to Julie Wilson. Julie, you want to stand up? She heads up product strategy and and patient advocacy at Septerna. I'm having a lot of luck here. Oops. Let me, I have to disclose I am an employee. I own stock in Septerna. So who are we? Septerna is focused on discovering and developing therapies targeted to G protein coupled receptors. We hope to usher in a new era of GPCR targeted therapies. We're founded by preeminent scientific leaders in this field. Our scientific advisors include Bob Lefkowitz, who won the Nobel Prize for his GPCR-related work, and we're headquartered in South San Francisco, California. Our innovation is based on our native complex platform, which I'll describe to you in just a little bit, and it is the engine to make these innovations happen, moving ideas from disease-modifying therapies from possibility to reality. Our first program is SEP-786, which is an oral small molecule agonist of the PTH1 receptor for the treatment of hypoparathyroidism. So why is targeting G protein coupled receptors so hard? Well, it's in the small molecule drug innovation area, there have been so many new developments that help us develop medicines for patients in need. But these innovations have largely been inaccessible to people trying to develop therapies for GPCRs. So really the root of it is because GPCRs are complicated. You can't just have that receptor protein by itself. It needs to be a part of a complex for it to be functioning. It's kind of like saying, I want to, improve an engine, but just taking a part of it, right? So the G protein coupled receptors are these complicated 7 transmembrane protein receptors that requires what it binds to, called the ligand, and also its associated G proteins. And all of these within what's called a lipid bilayer, or part of a cell membrane. So Septerna has been able to isolate and make these complexes and have them wholly functional, and they represent the biologic activity of the fully functioning unit. And by doing so, we can take these functional complexes and we actually create high resolution structures, which serves as kind of like a roadmap to identify novel binding pockets for chemicals and new drugs, and enables a smart structure-based design. We can also apply these to screen, and here I'm gonna emulate my inner Carl Sagan, billions upon billions of chemicals and physiochemical interactions, and we can apply what are called in silico or computationally based drug searches looking at billions of structures, and also DNA encoded libraries. So these are really industrial workflows, high throughput processes, so we can identify great drugs to what the roadmap had revealed itself, in the form of these native complexes. And then we could use structure-based design and optimization. With these candidates, we can identify what are the best drug-like features. And at Septerna, we've had just incredible luck using this process in identifying new drugs, and it often takes less than a year from the time that we've gone from native complexes to a drug that may be worthy to test in humans. So, the first disease that we decided to tackle was hypoparathyroidism. Now, the people in this room could teach me a thing or two about hypoparathyroidism. So, it's not worth your time for me to go over these. But what is just an incredible learning, and also observation from all of you, if you look at your little cards there, that we're in a veritable renaissance of innovative therapies for this disease. And I think, Liz, you mentioned a question about, are we going to suffer because of all these drugs? Actually, having developed many therapeutics in rare and non-rare indications, there has yet to be a disease that did not benefit from people developing options for patients. And here, every drug that comes to become available for patients adds to therapeutic options. Here at Septerna, what we are working towards is providing a therapeutic option for hypoparathyroid patients in the form of an oral small molecule therapy that is an agonist to the PTH1 receptor that could take the form of a tablet that's taken once or twice a day. So how do we do this? Now, I think I forgot who I was talking with before, but maybe it was you, Sissy. I'm not too sure, but yeah, I forget. But I think someone asked me, "Oh, wait, do you work on that drug that was licensed from AstraZeneca or something?" But I was like, "No. Well, we're working on 100%, full-blooded, in-house Septerna innovation. Where we are developing these drugs from scratch. And here, for the investigational drug we're developing for hypoparathyroidism, it started with this native complex that we talked about. Now, one of the things is why we targeted this receptor and this disease as our first candidate was it has been known to be notoriously difficult. Industry has been trying to develop small molecule drugs to do just this. And with the native complex platform, we've identified multiple hits and potential binding pockets that have heretofore never been worked on before. And with that, we applied the native complex structure-based drug design, as we had mentioned, and in about a year or so, we've optimized a candidate for great drug-like properties, and it led to an investigational drug that we called SEP-786. And we actually ran it through a battery of tests, one of which was a fairly well-established animal model of surgical hypoparathyroidism. It's when you take rats, and it's called the thyroid parathyroidectomy model, where they remove the parathyroid glands, and they develop hypoparathyroidism with chronically reduced calcium, and we treated with our agonist SEP-786. And on the right panel, you see the effect on serum calcium after single doses. And what you see is really what we consider drug-like effects. That means a dose-dependent increase in serum calcium, and we're able to normalize serum calcium within the normal range with single doses, and also to see how much we could push the pharmacology. We made a comparator to a long-acting parathyroid hormone peptide, and to push the doses all the way up until it's above normal calcemic range until you reach the plateau effect. So in this single dose animal study, so to speak, we're able to show the full range of pharmacology with this small molecule up to what is achievable with parathyroid hormone peptides. and with multiple dosing, that is sustained dosing twice a day, and we chose twice a day because in rats, the half-life was only 4 hours for the drug, and that means the amount of time it takes for half the concentrations to go away. Rats have this notoriously fast metabolism, so they chew up drugs very quickly. But we dose these rats twice a day over 28 days. And here in these little blue triangles, and you see this gray bar indicating what's a normal calcium range in rat, and that we were able to achieve sustained calcium control in these rats. But not only that, that sustained calcium control was achieved by its effect of our drug on the target organs. That is to say, we evaluated bone turnover markers, and to show that an increase in bone turnover markers, and also the effect on the kidney, we're able to see a decrease in serum phosphate due to phosphate excretion, and also an increase in active 125 vitamin D production that comes from the kidneys. So not only did we show a normalization of serum calcium with our investigational drug, but also that was achieved by its effect on the receptors on its target organs in the bone and kidney. So heading into human studies, there was a thorough evaluation in preclinical and non-clinical studies. And for my part, these were confidence-inspiring. The drug's metabolism and drug behavior, called pharmacokinetics, we evaluated these features in mice, rats, dogs, and monkeys. And the totality of this evidence suggests that the predicted human pharmacokinetics is such that SCP-786 would most feasibly be dosed once or twice a day. Also, we ran it through the usual battery of what's called GLP toxicology studies or toxicity studies. And it's a requisite part of drug development that we dose animals to excessively high doses as high as feasible, to make sure what happens in that setting so that we can navigate such things in humans. And what I'm very happy to say is that the toxicity studies showed that our drug was generally well tolerated after 28 days of continuous dosing. Now, the only thing that was really observed was what's called excessive pharmacology, which is when you have an on-target effect, the intended effect of your drug, but when pushed to excessively high levels, you saw higher effects. So high PTH-related activity is what we saw in toxicology studies. What is very reassuring is that we haven't seen the kind of what we say off-target effects, or the kind of things small molecule drugs sometimes have these liabilities that have an effect on the liver or other organs that are unintended, and our toxicity studies did not show any of those. So with this highly reassuring non-clinical evidence, first, good behavior in animal toxicity studies, and also animal proof of mechanism to show that we can actually address the surgical hypoparathyroidism in these animals. We went ahead and proceeded, and we just recently initiated our phase 1 human clinical trial that's initially in healthy volunteers. Yay. That's in healthy volunteers, and to evaluate single and multiple doses with the primary evaluation of its safety, and also the pharmacokinetic behavior, and also the pharmacology, the effect of this drug on serum calciums and other markers. And we anticipate sharing this information mid-next year. So this is also, and just as Patty had mentioned and Julie facilitated this visit, it is our plan for the full life cycle of drug development with SCP-786 to be conducted with patient focus, or more appropriately, with focus on people living with hyperparathyroidism and their caregivers. And also We are looking forward to a fruitful collaboration with the Hypoparathyroidism Association. And having developed multiple therapeutics in rare disease in the past, I can't tell you just how much the entire process is enriched by, by sharing the journey with people living with the disease and their caregivers. And at the outset, we're learning as much as we can about the unmet need and disease burden directly from talking with you. And we also expect and will be reaching out to you for your help in design of our clinical studies and with your input. And after the conduct of these studies, we expect to talk with you again and gather some feedback about how we could have done it better. But also, what's especially important is to get your feedback with the results of the data in hand to say, how do we interpret this benefit risk, of this therapeutic? And it helps us in terms of how we propose the eventual, approvals of the drug to be taken forward to patients, for real access, and then also to partner with patients and the healthcare for the advocacy groups with optimizing access to the drug and also with ongoing evidence generation. Advance. Thank you. So hypoparathyroidism is a beginning for us, and we are really intent on making it successful for you, the patients and the caregivers. But on top of that, it's just a part of our journey. We've started with hypoparathyroidism, but we have a deep pipeline of additional therapeutics across different disease areas, including immunology and inflammation, and other areas of endocrinology, including Graves' disease and thyroid eye disease, and also metabolic disorders. So with that, thank you so much for having us here today, and happy to take your questions. And Julie and I are here, and we look forward to chatting with you in the hallways. So
Okay,
Julie, you want to come up?
questions? We'll take one here first.
Hello, doctor. I have a couple of quick questions that might not be that scientifically wonderful because I'm just a layman. The first question that I had is, Septerna has started with trying to battle hypoparathyroidism. What was the motivating factor to choose that for Septerna? And my second question was, using this process, if it is affecting the ability to regulate calcium, what effect does it have on other, I don't know what you would call them, but things like phosphorus and magnesium and other such parts that come along with this problem?
Oh, that's a great question, and please don't excuse yourself because your question was amazing. And so first you asked why hypo-PTH, and the other is what do we think our investigational drug might address for the disease, right? Well, first, why hypo-PTH? One, because it had a long track history of being difficult to target for a small molecule. The other is, as a rare disease, it's something that as a small company can take the journey from beginning to end on our own. So we wanted to have, really our flagship initial disease that we target is something that we could take ownership for. Now, the other, in terms of what we are targeting for the therapeutic value of our drug, we actually designed the drug to hit a part of the receptor in such a way that we reciprocate the entire biology of parathyroid hormone. So the way we're dosing it, the way we'll target it, the way we're gonna try to match the pharmacokinetics is really is with the intent to replete the function of parathyroid hormone. To replicate the physiology and action of PTH in every respect.
Did you say in every respect?
In every respect. That's our hope now. And I think we have aspirations, and the design of the molecule is such that we hope the evidence will bear out that it could.
Hi, I'm Caroline. I had a question about, you said every aspect. So how are you going to manage variation in activity levels?
Well, that's a great question. Caroline, yeah, how do we manage the variability in activity levels? And well, that's one of, that's one of the advantages of an oral small molecule therapy. As I had mentioned, with our pharmacokinetic studies, we think that we could dose this drug once or twice a day, and we'll see how the human data bears out, if our predictions are correct or not. But what that also means is that we can hit steady state activity in a couple days. So with this drug, we can actually, I think that listening to you and listening to many of you, there are different types of demands. And I was talking with Dr. Khan earlier as well. Some people like to exercise in the mornings, and where it places a greater burden on their systems. So one, it's a part of the journey of this drug to talk more with what patients' unmet needs are, and to see if there's a way of dosing this drug that might meet you with those unmet needs. So I'm sorry I don't have a declarative answer because the drug is still under development, but I think it's really in partnership with you and our dialogue that enriches this drug development, evidence generation. But maybe, Julie, you want to comment a little bit about maybe how, some about our approaches in partnering with patients?
Yeah, so I think Patty already brought up our initial. We had just pretty much opened our doors not long after that. we were all scientists at the time. We're not anymore, but at the time, and when I say scientists, I mean biologists and chemists, not development scientists and statisticians, et cetera. And we brought them in because we really wanted our scientists to understand what patients go through. What is the patient experience? Why are we in the lab working so hard trying to find this drug? Needle in a haystack, I think you can kind of tell from how he talked about the process. And these guys came in and, like you said, our scientists were wowed. They wanted to know more, and I still get asked questions about it. We need to do it again, guys, so we're due.
Yeah. So we'd be happy to. Yes, and one of your scientists stopped us at ASBMR, and they remembered us from that talk and said, oh my gosh, I have so many questions still. And I was like, okay. But maybe you guys are the perfect example. There was a question earlier today about, the whole clinical trial process. You guys are our company that's come in the earliest in the game for us. And a lot of patients don't understand. So the question I'm referring to is the one that was, what did you mean by clinical trial versus phase 1, 2, 3? You're the first time we've been this involved and had somebody in front of patients that is just heading into phase 1, and you were just in preclinical. Maybe could you talk about how that timeline works and not too much detail, but just a high level of how that works and how it is going into a phase 1 and what it looks like. What is your anticipated phase 2?
Yes, well, yeah, you might not be talking with the ideal person for this because I generally see the rules and try to see if there's ways I can break them.
I'm perfectly okay with that.
Well, what I mean by that, well, that could come across in a really bad way. But what I mean by that is, I've been in rare disease, drug development for a while, and what I've discovered are that really, every day counts, every day, and that when you can bring a meaningful therapeutic to people living with disease and their caregivers, with that in mind, that every day counts, you think about ways that you can innovate along the way. So I actually don't think of it in terms of phase 1, 2, and 3, because I've leapfrogged phases all the time, right? It's all about evidence. What questions are you answering? And so maybe just to share with you, from a drug developer's standpoint, the purpose of drug development is to understand the dose or exposure relationship with benefit and risk. That's what you're characterizing. So when we say phase 1, that is, usually when you dose the drug for the first time in people, in humans, and you could decide whether, depending on the situation, whether it's healthy people or people living with disease, and it's very situation dependent. But that's when you first evaluate the safety of the drug as you push the doses. And when you see a dose-like effect, that's when you say, you've got a drug. That's what defines a drug. It's a dose response. Anything could be a drug, right? But if it doesn't have a dose response, it really isn't one, So that's when you say, hallelujah, we have a drug, and then that's where the journey starts. And then you start building up your evidence for a justification of how you dose that drug going into what we call a pivotal or registration study, right? Whether or not you need a phase 2, it's all dependent upon the situation. There's actually no regulatory definition for phase 2. It's if you need something in between to help fulfill that evidence requirement before you go into a pivotal trial, right? In ultra rare diseases, sometimes you can just have one phase of development, keep amending a phase 1 from first in human all through pivotal, and then make that your registrational study, right? So that's why, for me, there's no rules, right? It's about evidence and how you can most quickly get adequate evidence to get your drug to move forward. And also, what is confidence inspiring for health authorities to review the package and say, oh, this drug is safe and it has an adequate benefit risk to give to patients, right? And it's called, for the US parlance, it's preponderance of evidence of efficacy, right? So yeah, for me, so there are kind of set definitions of phase 1, 2, and 3. And sometimes when you're trying to figure out what the best way to get the therapeutic to patients as quickly as possible, those lines get blurred. But that might be a bit more complicated an answer than you asked for.
No, I think that it helps them understand. I think that's great. As a follow-up to that, can you tell our patients, if I might have missed it because I had to step out for 2 seconds, but do you have an idea of what this phase that you're in right now, because you guys are getting ready to dose your first human, or you have just dosed it in Australia? Did I understand that correctly?
That is correct.
You did dose your first human in Australia. What does this current timeline that you're in, what does that look like? Is this a 1 month, a 1 year, a 5 year? Because patients don't know. How long do these things take?
Oh, yeah. Well, so there's a limit to what I can disclose at this time, but what I can say is that we are definitely in the first in human study in phase 1 of development, and we are evaluating what the safety and pharmacology is of the therapeutic and in single and multiple doses, and that we will be sharing those results mid next year. And I think we'll be better prepared at that time to share what the rest of our program might look like.
Okay, and then one more real quick that just came in online. It says, how long, I don't even know if I understand this, how long is the intracellular action of this drug?
Well, how long is it intracellular action? Wow, this is complicated biology.
I know, I was like,
Well, I think so. I'm interpreting that question as so a drug's effect, right, is evaluated by both what's called pharmacokinetics, or the concentration, or how the concentration of the drug behaves in blood, like how the concentration rises and falls after you've taken it by mouth, right? And then there's the effect of that drug. Sometimes the effect of the drug can be longer lived. That is, the effect on the cell itself can have, longer lived actions on calcium, right? And so I think, in terms of what we've seen in animals is that the effect on calcium is a bit longer than what's in the blood, but not too much longer. So I think what that helps is that we can actually use how we dose the drug by mouth as a good indicator of how we're achieving steady state and so forth.
It's a great question, though, you asked about the timing of the phases. I mean, there is a standard 1, 2, 3. A lot of drugs do, the majority, I think. And the time, though, really differs. So if you think of something like osteoporosis, where you're trying to measure whether a drug keeps the bone from breaking down over time, those trials are very, very long. And then hypopara, wouldn't you put that on the shorter end of time in general?
Yeah, maybe to help you walk through the journey of what it's like to develop these type of therapeutics. So you kind of usually classify them as, one, drugs that are intended to treat those that are immediately life-threatening, right? Versus those that are used to treat chronic conditions that are not immediately life-threatening. I raise that point because if you're treating a chronic illness, that I would classify hypoparathyroidism as a part of the evidence that you have to say is that your drug is safe and it works for at least a year for however many patients that is acceptable to the health authorities, that is FDA and EMA, right, and other health authorities where you want the drug to be available. So usually one requirement in that drug development process is to say that, okay, we took X number of patients with the disease and we treated them continuously for a year, and then that's used to fulfill that minimum criteria for safety for a chronic drug. So we have to kind of figure out where that falls in, and so that tends to be the, quote, limiting part, is that you have to at minimum satisfy that part in addition to what might be required timeframes to show efficacy. Because you can say it doesn't take very long to show that you're feeling symptomatic benefit from hypoparathyroidism. So there are all these, they're called statutory requirements for when you're developing drugs, and that tends to be one that we think about a lot in advance, is how can we build in that 1 year of safety, that's the sliding part of that timeline. I don't know if that answers your question, but
It
okay.
did, it definitely answered my question. I want to make sure, is there any more from the audience? Okay, yeah.
I'm wondering why you picked Australia.
Liz, it's up to you to ask the hard questions. No, it's actually there are many great places to do phase 1 first-in-human trials. We conduct them in what's called professional phase 1 units, and especially for small molecule drugs, you have to have a very, very careful time course, and it's like it's almost like being in like NASA, right? They have these like time clocks everywhere, and everybody has on a very regimented, blood collection and markers and blood pressure, all these things, and with small molecule drugs, they tend to require a very precise schedule to do these, and it has to be done in what's called a professional phase 1 unit. Usually, average clinical trial centers really don't have the precision to conduct these studies. So there are many of these, really good ones in the U.S., in the U.K., Australia tends to be one of those units that can conduct these trials very well. That I've conducted phase 1s at this particular unit before. So for these phase 1 units, it tends to be, there are multiple, reasons for selecting which unit. The primary reason is it's a unit that that we have good experience with, and they produce high quality information. And the other is it just happens to be some features of how Australian submissions work, that it can actually start a study 2 months earlier than one that is conducted in the United States. So, we wouldn't cut any corners or anything, but I think it's a combination of being able to get the study started earlier, and also, because of the high quality information they can provide.
All righty, well, thank you so much. It's so great to have you guys here.